Thank you very much for the kind introduction and for the invitation. It's a pleasure to be speaking at your seminar, and it's nice to see familiar names in the audience. Hi, Jean. Hi, Jamie. I mean, right. So you all know what quantitative MRI is. I'm just going to do like a brief introduction about the rationale for this talk. So it's basically an umbrella term for many techniques that use MRI. Its main purpose is to generate maps that reflect the underlying tissue microstructure of the central nervous system. We can also probe neuronal activity with MRI using so-called functional MRI. It's based on the bold signal. We can also look at metabolic concentration, and ultimately all these quantitative markers are valuable to help in the monitoring of diseases, for improving diagnosis and prognosis, and also test the efficacy of therapeutic drugs. But while QMRI has been widely used in the brain for more than 2 decades, it painfully makes its way to spinal cord application. So why is that? Challenge number 1 is related to the dimensions of the structure to image. And as you can see, the spinal cord is a small structure, which is about 1 centimeter diameter. And it therefore requires high spatial resolution. And as you know, the better the resolution, the lower the signal-to-noise ratio. So to overcome this problem of sensitivity, one solution is to get better hardware, and especially receive coils that's with more elements, smaller elements that are closer to the region that we want to image. And the second approach, which could be combined with better coils, which is a little more expensive, is to get a more powerful magnet. So I'm gonna show you a little bit about our recent research where we combine 7T MRI with better coil geometries, and that's so it's a work that's done in collaboration with Kyle Gilbert at the Robarts. So Kyle was involved in the design of the transmit coil, which consists of 8 electric dipoles that are located around the head and the cervical cord. And we designed and built the receive part of the coil, which is 20 receive elements, which are in a soccer ball design, and that covers the lower brain, the C-spine, and the upper thoracic spinal cord. And what you see in red is additional shimming components, because one of the purpose of this coil, for some of you who know, is to convert this receive coil as an AC/DC coil, where we can also do local shimming. So I'm going to say a few words about that later. So that's the design of the coil. It's compatible with the Terra system and the 8-channel transmit system. So we can adjust the height in order to be close to the anterior part of the neck. And that's the built coil. And given that we got satisfactory results, we actually built a second prototype, the exact same replica, and it was shipped to Rob Berry at the MGH Martineau Center. And so his team is using the coil for fMRI applications. So it's nice to have the same coil at 2 different sites to be able to assess also replicability of the results. A brief overview of the coil performance in terms of receive. Here you see a flash image with the sagittal view for each of the 15 posterior elements and the 5 anterior elements. So there is a good contribution for each of the receive channel. since the coil has 8 independent transmit elements, we can also control them independently and play with the static RF shimming, where the magnitude and the phase could be optimized in order to, for example, obtain a perfect or like a satisfactory homogeneity in the transmit field in the region of interest, which in this case would be the cervical spinal cord. So if we look at the B1 plus maps and quantify the signal along the spinal cord, so it's just a different color, but it's essentially the B1 plus map that is put into the this nanotesla per volt color map. We see that there is better homogeneity that we can also quantify along the cervical spinal cord when we adjust the magnitude and the phase of each of the elements compared to the default CP mode that was set for this particular coil. And using these optimized transmit magnitude and phase for each element, we also run an MP2 range sequence where we quantify T1 along the cervical spinal cord. And we observe that there is a more stable computed T1 because of the like the reduced effect of B1 plus inhomogeneities compared to the CP mode. So this opens the door to applying some of the QMRI techniques in the spinal cord at 70 with fewer bias related to transmit inhomogeneities. So if we go back to our challenges, that was about the challenge of improving the resolution or the SNR. The challenge number 2 that we face when we do spinal cord imaging relates to something called susceptibility artifacts, which you might have heard of if you're doing also brain imaging, especially at higher field. So here I'm showing an image of a diffusion tractography. So for those who are not familiar with it, diffusion MRI is a technique where we use the gradients not only to get an image, but also to encode the diffusion of water molecules. And from this encoding of the diffusion of water molecules, we can then reconstruct the pathway of white matter tracts inside the central nervous system. So there are like a lot of people are familiar with those images in the brain, but we can also reconstruct the tracts in the spinal cord. And what we seem to observe here is that there seemed to be a complete spinal cord injury at around C6 vertebral level. But in fact, I was the subject. I was taken a couple of years ago, and this interruption of tracts is only artifactual. And in fact, if we correct for the distortions on the EPIs that were used to acquire these diffusion-weighted scans, we see that the tractography then, continues below the C6 level. So that also raises some concerns about those advanced techniques that one needs to be extremely careful when interpreting results of tractography or any other quantitative MRI techniques, because the artifacts could lead to a wrong conclusion. So now, what do we do with this? So there are ways to mitigate this artifact with in brief, the idea is to acquire the EPI faster to reduce the amount of distortions, and there are different techniques with EPI to do that. Another approach, which could be complemented with better EPI sequences, is to do a better shimming, and so it really addresses the problem at the source, where we are trying to correct or like to mitigate for these B0 inhomogeneities. So I'm going to talk a little bit about shimming because that's a dear topic of mine. So if you look at a magnet at an MRI scanner, you get you see the magnet right? I mean, you don't see it because it's covered, but you imagine that there's a very big, powerful magnet. That's the famous part of an MRI. There are less famous parts. One is called the gradients, which is used for, for encoding and to get an image, but also for diffusion MRI experiments. And the even less famous are the shim coils, which are additional coils that are used to modify the static B0 field over a desired region. And for example, here you see a phase field map that shows a poor homogeneity around the cervical region. It's poor homogeneity because the phase is not zero, and it's not the same. It varies across the cervical spinal cord. And by inducing currents into those shim coils and gradient coils, we can make sure that the region that we are interested in is more homogeneous in terms of the B0 field. So that's shimming, that's active shimming. But shimming in the spinal cord can be hard for two reasons. The first reason is that in the spinal cord region, there are a lot of small spatial scale inhomogeneities that are caused by closely spaced structures with different susceptibility profiles, such as the cartilage, the bones, and the lungs. And the default shim coils that are integrated into the MR systems can usually achieve up to third-order spherical harmonics. The spherical harmonics are essentially the polynomial functions in 3D, and they are not sufficient for compensating these spatially complex geometries. So we would need to build shim coils that have densely packed shim elements and that are close to the region that we want to shim. Okay, that's specs number one that we want. Another problem is that these inhomogeneities are not frozen in time, and as the participant breathe, the B0 field is also distorted dynamically across time. And this causes all sorts of artifacts, such as ghosting on anatomical GRE scans. So we would need also to modulate the shim currents in real time along with the respiration, right? So based on those observations, a couple of years ago, we got in touch with resonance research, Piotr Starevich, based in a city close to Boston, and they designed and built this 24-channel shim coil that is put on the patient table, and that can run independently from the scanner. So each element that you see here can produce a field. And so the idea is to characterize the B0 field contribution in the Z direction from each of these shim elements. And then we can use that to fit any field map in order to compensate for the residual inhomogeneities. So on top of this shim coil, there is an RF coil, transmit and receive, because we could not use the transmit coil with that heavy copper shim coil. So we had to use another RF coil, and on top of this lies the subject. Here is Ryan Topfer, who did his PhD using this technology. So that's the hardware, and we need to orchestrate it. So the setup works that way. So we put the shim coil on the patient table, then the RF coil, the subject on top of it. The shim coil is controlled by a dedicated amplifier that sits in the equipment room. And we also record the respiratory trace from the subject, and that goes into custom microcontroller that goes into a laptop, and that laptop is connected to the single console in order to acquire the B0 field maps as they are being reconstructed, and in parallel also having the respiratory trace. So it's very similar to the Van der Gelderen paper back in 2007, where they did this in the brain. Here, the idea is to do that on an external shim coil, And from, by acquiring the respiratory trace and the bezier field map, we can then say, for this particular, phase, like location in the respiratory phase, we want this combination of shim currents. And so this information is sent to the controller in real time, and the shim is modulated in real time. So I'll show you some results of this experiment. What you see on the right is the respiratory trace, which was accelerated for the purpose of the presentation. And what you see in the middle is an EPI, so it's a sagittal EPI, okay, that is acquired with a relatively short TR. I think it's about maybe 200 millisecond TR, something like that, to really appreciate the variation of the bezier field across time. And there's an apparent motion of the cord, but this motion is not due to the to the chest or to the body motion, but it's purely artifactual. The bezier fluctuation induce a shift along the phase encoding direction, and that's what makes the spinal cord move, so to speak, right? So when we activate the real-time shimming, the image is more stable across time, because this real-time shimming compensates for the bezier fluctuation dynamically. Okay, and to convince you that this apparent motion is not due to the subject moving, we've also acquired GRE scans, so non-EPI, which are much less sensitive to these bezier variations. And you see that even if the subject is breathing, we don't see a motion of the spinal cord, right? So that was really an effect of the bezier change. We can also go a bit further and combine real-time with dynamic Z shimming. So dynamic shimming relates to the idea of setting a particular shim coefficients per slice. So that's valid and that's variable when we do 2D imaging. And the idea here was to combine dynamic shimming, or slice-wise shimming, if you will, with the real-time shimming based on the respiration. And so for that, we did not use an external shim coil. We only relied on the scanner hardware, and a gradient blip was calculated based on the static positioning of the slices, as well as the location in the respiratory trace that we get from the respiratory bellows. And in that case, so that was done on the Siemens scanner. So all this was integrated into the IDEA environment, in a sense that we could fetch the respiratory trace from the Siemens bellow and incorporate that for the calculation of each gradient blip. So that work was done by Eva Alonso-Ortiz, and it is now available. I think it's been accepted a couple of days ago, so you can look at the Neuroimage article on this topic. So we talked about the hardware, we talked about the pulse sequence programming, but there is another aspect that is important if we want to do these experiments. It's software. That's really the glia of all this. Without software, we cannot do anything. So for the past few years, we've been working on an open source software package called Shimming Toolbox, which runs as a standalone Python library on a laptop that is connected to the MRI console. So it gets the DICOM immediately after they are reconstructed, converts them into a nifty BIDS format. BIDS is an acronym for brain imaging data structure, and it's just a way to name the files so that we can use BIDS compatible applications to process the data without having to rename them on the fly. So the Shimming Toolbox runs as a plugin into Facialize, allowing users to visualize the images, generate masks, QC the masks, and QC the results of the shimming. And so it computes B0 field maps, and it captures the respiratory trace in real time. It can also create masking tools, so leveraged by FSL, BET FSL for the brain, and spinal cord toolbox for the spinal cord. It can generate coil profiles for custom coils, but also spherical harmonic-based coils for 3T or 7T scanners. It does the fitting for each coil element. It can also combine elements, so we can run shimming experiments by combining external coils with the gradient and shim coils of the scanner. Sorry, there seems to be a lot of police activity on the street. And then those coefficients are sent back to the laptop that writes a text file, and the text file is read by the pulse sequence of the scanner, and then it can then consider those coefficients for the advanced streaming. More details in this recent MRM publication from Alexandre Dastou. Okay, so we've talked about resolution, SNR, coils. We've talked about shimming. Another challenge, quite different, is if you remember my first slide showing the different QMRI methods. In fact, I was only showing you some of the QMRI techniques that exist. And every year at ISMRM, new sequences and new techniques come up. So if a researcher not familiar with QMRI was to start a project involving spinal cord imaging or any organ imaging with MRI, what sequence and parameters to choose from? And moreover, in order to address the challenges previously mentioned about need for high resolution, low SNR, susceptibility artifacts, one has to carefully select the acquisition parameters. And there is a lot of them. So picking the right methods and acquisition parameters can be overwhelming for newbies in spinal cord imaging or any type of MRI imaging with quantitative techniques. So with that in mind, the spinal cord imaging community has been working for the past 5 years or so on a standardized acquisition protocol for spinal cord quantitative MRI data. And this protocol is called the Spine Generic Protocol. It involved 42 international sites that have been involved in this optimization and validation. And so importantly, the protocol is available for the 3 main vendors, and it is using product sequences only. So you can see more details in the Nature Protocols article that describes the protocol. You can download it on the GitHub page, and it's constantly updated, with the new VIDA platform or like the XA30 and so on. There are like new protocols that need to be updated. And so they are, usually if you have a problem, if you don't find your version, post an issue or contact me, and then we'll make this happen. So in brief, the protocol includes a 3D T1 weighted scan that includes the whole brain, and a higher resolution T2 scan as well for cord atrophy measures. There is also a 2D high resolution gradient echo scan, multi-echo gradient echo, to have a good white and gray matter contrasts. And seeing the gray matter in the spinal cord is important because it enables us to segment it and then get some measures of atrophy of the gray matter, which could be relevant for disease such as ALS. The protocol also includes diffusion-weighted sequence to compute metrics that are sensitive to demyelination and axon degeneration, as well as magnetization transfer protocols to compute the magnetization transfer ratio and MTSAT. To maximize reproducibility, the protocol includes a comprehensive document with a step-by-step procedure that is directed towards MR technicians that tells, how to position the patient, how to position the slices and the SAT band, depending on the type of vendors and license that you have. And in order to validate that the protocol could be reproduced at different sites, we've also acquired data, a lot of data, across different sites. So there is one data set of a single subject acquired at 19 sites internationally, and another data set for multiple subjects that were scanned across 43 sites. As I mentioned before, the BEADS data structure was used to organize these data sets, and they are downloadable using GitAnnex or DataLad for those who are familiar with it. It's essentially a Git-like technology to be able to version track the data sets, but it's more adapted to big data compared to Git, which is only used for code. So the way we organized this multi-site collaboration, it was a bit of a logistic challenge. So each site acquired the data, anonymized them, converted them to bits using scripts that we provided, and then the data were sent to the managing team, which added some quality control assessment, some data processing, and the data were pushed onto an AWS server, and the metadata and the data set is listed on the GitHub repository, so that everybody could, with one line of code, access the data. It's actually 2 lines of code. So what do we do with this data set? Well, let's look at it. Let's process it and see what kind of metrics we could get out of that. So first of all, let's look at the T1 weighted scans. So these are the ones from all the subjects, and as you can see, the field of view placement was well respected across all the different sites. So we were quite happy with that. And then what you're seeing here, so it's a bit of a complex slide, but on the left, it's a sagittal view of a cervical cord that was segmented. So the segmentation is in red. And then we identified the vertebral levels, and we computed the average cross-sectional area from the spinal cord segmentation and averaged it across the C2 and C3 vertebral levels. And this metric is very useful in disease such as MS, where cord atrophy is a good indicator of the severity and type of phenotype of MS. So this cross-sectional area was computed for each participant. So each participant is represented as a red dot on this bar plots, and the color coding is based on the vendors. So black is for GE, blue is Philips, and green is Siemens. And you see that we also had access to different models from each vendor, right? So overall, the intra-site, so when we look at each different site, the coefficient of variation was below 8%, which likely reflects the variability across subjects, the anatomical variability across subjects. But when we look at the within vendor and inter-site coefficient of variation, that was below 4.2%. So we were quite happy with that. Another metric we wanted to look at is the magnetization transfer ratio computed in the white matter across the C2 and C5 levels. The intersite COV was below 2.5%, so very small, smaller than with the CSA, which goes along the knowledge that the MTR in the white matter is more stable than the cross-sectional area across participants. And The two red crosses are there because the two sites did not use the suggested parameters from the protocol. They used a different TR, and that affected tremendously the MTR result, right? So they were excluded from the statistics. And finally, the results from the diffusion tensor model showing the FA value. And again, relatively small COV across sites and within vendor that is below 4%. So there are more results, which I don't have time to show here, but they are available online. So if you Google spine generic protocol, you can access the page, which is constantly up to date. You can see some of the data sets and interact with them for the different contrasts, the T1, T2, T2 star, and the diffusion weighted scans from different sites. And you can also have an idea of the analysis pipeline that was run, as well as the results that I just showed you. And what's nice is that you can interact with them. That's built with a Plotly, so you can have the values. You can see the different metrics that were run for this data set. So there's a lot of things to play with. So let's do a quick summary of the different challenges. One was the SNR and the better hardware that is needed. Challenge number 2 was focused on susceptibility artifacts and shimming. Challenge number 3 was about this spine generic standardized protocol and open access dataset. And challenge number 4 is about data processing. Because quantitative MRI techniques require advanced image processing methods. And for the past 20 years or so, several software packages have been developed to process brain qMRI data, such as FSL, SPM, AFNI, and other popular methods. But unfortunately, until recently, there was no dedicated package for the spinal cord. And the existing package for the brain cannot be easily adaptable to the cord because atlases and image features are different. So that triggered the development of the Spinal Cord Toolbox, or SCT, which is still an active development in my lab. And in brief, it features specific segmentation tools, atlases, a framework to do atlas-based registration, and various tools for motion correction and so on. So I will focus on two particularly important aspects of SCT. The first one is the template, which the latest is called the PAM50 template. PAM stands for Polytechnique, the A stands for Aix-Marseille Université, and the M stands for the MNI. It's a collaboration between these three groups. And 50 because it's based on 50 healthy adults. So there are T1, T2, T2 star contrasts, also a probabilistic atlas of the white, the gray matter, and an atlas of the white matter tracts, so that quantitative MRI metrics could be quantified within each of these sub-tracts. Another important set of tools which are in continuous developments are the segmentation tools. For the past few years, we built those tools on deep learning, and there is one for segmenting the spinal cord that includes, so the model includes patients with MS, spinal cord compression, NMO, ALS. So we are trying to make those tools robust to the different pathologies and not only trained on healthy controls. Segmentation tools also for the gray matter from ex vivo and in vivo scans, as well as tools for segmenting pathologies such as MS lesions and also tumors and different parts of the tumor. So that was a recent work by Andréane Lemay. And everything is powered by another toolbox called IvaDomed, which I'm not going to go into, but it's another active project to train models of medical images. So I'll show you to finish my talk. I'm just going to show you a few applications of SCT. One that was an interesting project, multi-site project, where we created a probability map of MS lesion distribution in the cervical spinal cord. Ironically, it was published in the journal called Brain. And this mapping can be used to study a potential signature of MS distribution across the various phenotypes. And so what's nice in having this probabilistic map in a template is that we can then interrogate the distribution within different sub-regions of the cord, for example, the gray and the white matter, and see if some particular phenotypes have a signature in terms of distribution of lesions in the white versus gray matter. And we could also use these atlases to look at the correlation with the disability scale. Moving to another disease, amyotrophic lateral sclerosis. Here, SCT was used to segment the gray matter and look at the potential for differentiating patients versus control. So that's something that was already known. But what's interesting is that these patients were followed 1 year after prognosis. And we looked at the prognosis value of the gray matter atrophy to get additional predictor 1 year after the onset. And another pathology is spinal cord compression, non-traumatic compression. That work was done in collaboration with the group of Michael Fillings at Toronto Western Hospital. And here, SCT provides tools to analyze the shape of the spinal cord in the axial plane and can output metrics not only of the amount of compression, but also in aspects of ellipticity or anterior-posterior diameter, right-left dimensions, how the cord is turned at the compression site. And Alan Martin notably showed that combining those different metrics could bring more insights into the severity of the compression. Stroke is another application where SCT was used, and the idea here was to look at diffusion MRI to assess the integrity of the brainstem and the cervical spinal cord. And the track-based analysis helped identify altered sensory motor pathways post-stroke in these patients. And finally, I like this application because it's a combined modality between PET and MRI. So the standardized uptake values, or SUVs, were extracted in the spinal cord at specific vertebral levels based on the anatomical MRI after registration with the PAM50 template. And so these results implicate immunoactive response at multiple levels of the nervous system as a potentially important and clinically relevant mechanism in human radicular pain, and suggest that therapies targeting immune cell activations may be beneficial for chronic pain patients. So to conclude this talk, we covered hardware aspects, focusing mostly on 7T technologies, talking about coils and shim technologies and how this could push the limits of spinal cord applications at higher field strength. We talked about a standardized protocol, which main purpose is to promote the replicability and the dissemination of knowledge for spinal cord QMRI applications. Hopefully it will minimize the variability of spinal cord QMRI results and implementations, and it's already implemented in at least 50 clinical sites and used in multiple initiatives, including the one a pan-Canadian study called CanProCo, which is led by Ji-Won Ho at University of Toronto. And it spans multiple sites across Canada, and its main purpose is to study MS patients. So the spinal cord imaging part relies on this generic protocol. From this generic protocol, we also created an open access database, which could be used to generate normative values in this healthy adult population that could be compared with pathological cases. And also, this public database is an interesting sandbox for improving the automated segmentation models. And finally, the analysis tools promotes reproducibility for more transparent analysis. And I love automated pipelines and batch scripting because it's really a way to make sure that results that someone presents can be reproduced on other systems 5 years after the fact. And there is also remaining aspects, which is about communication, and in particular, discussion about unmet needs that needs to happen between physicists, clinicians, and MR vendors in order to make sure that, if there is a good idea that physicists are talking about, it could be implemented by the MR manufacturers and then benefited from by the clinicians. And in order to promote these kind of communications, we've been organizing workshops about spinal cord MRI since 2014, every year during ISMRM, except during the COVID time. But these workshops are a great opportunity to meet and discuss these methods and push the field forward for better implementation and adoption of these techniques. So if you would like to be on the mailing list of the events, please visit www.spinalcordmri.org. And with that, I would like to thank everybody in the lab who contributed to this, to the science, the collaborations, and also the funding sources. And thank you very much for your attention.